Notre prochain orateur sera donc James Farquhar. Quand on a réfléchi au titre de l'après-midi scientifique, les isotopes du soufre étaient un des sujets retenus et on s'est dit que c'était peut-être l'occasion rêvée de faire cet après-midi scientifique sur les isotopes du soufre parce que James Farquhar est en sabbatique en France sur une chaire franco-américaine Fulbright Tocqueville. Il a passé 6 mois au laboratoire en magma et volcan de Clermont-Ferrand et l'autre partie de son séjour à l'IPG de Paris. Et donc James est américain, il travaille à l'université du Maryland. Il a fait une thèse au départ au Canada, à l'université d'Alberta, et puis 2 ans de postdoc à la Carnegie de Washington. Et c'est en postdoc toujours chez Mark Timmins à la même époque que Joël, et les deux compères se retrouvent, que la carrière de James a vraiment décollé. Non pas qu'il n'avait pas des papiers à nature, mais il en a eu vraiment beaucoup avec son stage postdoctoral, et notamment pour sa découverte des fractionnements indépendants de la masse du soufre dans les sédiments archéens. Alors, on n'a pas demandé à James de parler de ça aujourd'hui parce que c'est un sujet qui a été largement traité dans nos différentes AMS, et on a plutôt profité de sa présence pour lui demander de nous parler de réduction du soufre par les différents types de métabolisme et les effets isotopiques associés. Et donc ça, c'est la carrière de James ne s'est pas arrêtée aux sédiments archéens. Il s'est vraiment concentré sur l'isotopie du soufre, qu'il a vraiment appliqué à tous les domaines. Et je pense qu'il a fait des travaux très importants en cosmochimie sur les différents types de météorites, mais aussi sur Mars. Et un autre volet, parmi tant d'autres, ça a été d'étudier les, les, les effets isotopiques liés à la biologie. Il a formé beaucoup d'étudiants, dont un qui brille, puisque c'est Dave Johnston qui doit être dans la liste des co-auteurs, qui est maintenant professeur à Harvard. Et je crois que ses travaux ont été initiés dans le cadre de la thèse de Dave Johnston, qui faisait une co-tutelle, à ma mémoire, avec Duncan Field, donc au Danemark. Donc merci James de parler d'un sujet un peu plus inhabituel, je crois, pour toi. Mais merci encore. C'est à toi.
Okay. Don't touch anything. Okay. Thank you. Thank you very much. I have what I hope is an interesting and fun talk. It's a different sort of talk. Some of which I will discuss is well known, and some is not. I thought I would give a little bit of background and context first. This is a picture up north of here. It's off of Mont Saint-Michel. And if you were to go out in those muds and dig down into them, you would I have to hear myself. Yeah, I can try. Yes. You would smell a characteristic smell of rotten eggs, which is sulfide, and it's saying something about the ecology of what's going on in the mud. Okay, that's there. If you went a little bit closer to where I live, so this is the Severn River in Maryland, and you went up into this bay here, which is Round Bay. You note that in the summer times, and if you dug down into the mud, you also would smell this sulfide-rich smell. And this is Zara Mansoray, and the mud itself has this blue-black color. It's iron sulfides, and it's something that is biologically produced. Okay. This metabolism also is present in the oceans. It's present worldwide, and it accounts for about 50% of the carbon mineralization that occurs in ocean sediments. And if you think about it in terms of a balance, the production of sulfide and the loss of sulfide from the oceans and the sediments accounts for about 30% of the oxygen balance that is produced. Okay, so the air that we're breathing today is in large part balanced by this. So it's something that's not trivial. It's something that's important. It's done by little organisms. And if you look back in time and you wanted to know a little bit more about it, this is a plot. Again, this is just the delta 34S versus age, present to past. This is the isotopic composition of sulfate and average. This is an average of the sulfide. It's actually, in this case, it's calculated through a model, but if you went through and did a running average, it would match very close to this. And you see here that the difference in the 34S between the sulfate and the sulfide appears to change through time. And we can get to this several different ways. All of this relates to the role of these organisms, and it relates to the way that their metabolisms are operating. So what we've wanted to do with these organisms is understand how the metabolisms operate and understand what are the controls that potentially determine the variability between sulfate that's feeding the organisms and sulfide that's produced. Now, this diagram here has many more levels to it. There are all the environmental levels which this goes through, but a lot of this could in fact also be related to the organisms, and people have suggested that. So thinking about the organisms and what they do, this is a genomic reconstruction of some of the different metabolisms and metabolites in a sulfate reducer. And you see there are various different pathways given by the arrows and various different enzymes and processes that go on. What's highlighted in here in the non-shaded part is the sulfate reduction metabolism of this organism. And we'll look at this in a little bit more detail, but you see there's evidence for transporters across the cell wall. There's evidence for production of different types of compounds like this adenosine phosphosulfate. There's evidence for enzymatic conversions to sulfite, and then reduction through a 6-electron transfer ultimately to sulfide that gets transferred out of the cell. And it's the whole set of these things that controls the isotopic effects that we see. Okay, this is also, it's an energy producing metabolism, so it depends on the environment, and it reflects how the organism is functioning. Now we'll look at that in a little more detail, and I'm going to show you a few aspects about this quickly through what this metabolism is doing, and then we'll look at these again. But this is a model of the metabolism. You have sulfate outside the cell. This would be the cell wall. Sulfate inside the cell in the cytoplasm, going to this adenosine phosphosulfate, then to free SO2 or S4. It could be bisulfite. And then it goes through a desulfite reductase enzyme, which does a lot of this and ultimately produces the sulfide. And in this, we have several things that are important that I want to talk about. And one has to do with the role of transport or transfer of sulfur from each of these pools. So from sulfate outside the cell to sulfate inside the cell. And in those cases, those are physical pools, but you could actually have it could be essentially a probabilistic pool. If you have something that's going on down a channel, you may not have a distinct pool that is in the sense of a large physically separable pool, but it's a whole bunch of channels that are operating in the same way. These things could go backwards, and that's one of the questions that isotopic geochemistry or isotopic chemistry has, I think, provided some key evidence for. There were a variety of lines of supporting evidence, but nothing really nailed it down. There also are various transformations. So if we goes to this APS, it forms the APS. The APS itself is reduced to sulfite, and this is a picture of the APS. It's an organic structure here with some phosphates, and then there's a sulfate here, and this thing gets inserted into a channel here to an active center here. It's a flavin active center, this FAD, and basically it docks in here, and then some sort of magic happens where their electrons get transferred, bonds get broken, and this thing then comes out and it releases sulfate. These enzymes can be several hundreds or thousands of atoms, and they are really pretty incredible how they allow for chemistry to occur that might not otherwise occur by changing the reaction conditions. You also, moving further down, go into this desulfite reductase enzyme. This thing has, it's a large enzyme, tens of thousands of atoms. It has two major units here. They would each look like this coupled together. Each one of them has an A, B, C unit. The C is the one in green here. It has an active center, which is sort of pictured here, looking in through the bottom of the thing here. And the sulfite goes in through a channel. I think it's about 9 angstroms wide, and goes in here and then docks here. And chemistry occurs. It converts the sulfite, which is S4 to S2 to S0. That S0 then is connected onto the C subunit, and then that C subunit undocks from the enzyme, floats around inside the cytoplasm, goes to the cell wall, and completes the reduction to sulfide. Okay, so it's a relatively complicated process. And so you've got the possibility of reversibility. You've got the possibility of enzymes. There's one other level that I want to look at, and that we'll look at, which is if you look at a species like sulfite or like sulfide, it speciates in solution. So under high pH conditions, sulfite is stabilized, the SO3, and this is the region in which these organisms, the pH of the cells, and under low pH you get the bisulfite. Same with the sulfide, you get sulfide H2S and then bisulfide up here. The pure S2 is off the thing here. But the speciation, how these things are protonated, and actually it gets even a little more complicated than this, because where the proton fits on the species makes a difference in the isotopic effect. And these things have different ways of partitioning isotopes between them, and all of those things can enter into it. So I want to just show you some of the considerations and show you some of the thinking that we have been doing that relates to these different steps. And the tool that I use is isotopic geochemistry. This is our old fluorination line, measuring, and we'll look at the 4 sulfur isotopes, mostly looking at sulfur 32, 33, 34, not so much 36, although we do consider that. And then we also use oxygen as a tracer in one of the examples that I give. We use the delta notation, so the variation it's a doubly normalized ratio, deviation from that for 34 to 32 relative to a reference. And then we use the capital delta notation just as Joel did, which is it's the measured value minus the predicted value. And we're not looking at a mass independent effect in the examples that I'm about to go through, but we do see an effect that can be seen. It's regular, it's something that can be understood, and it gives us information. Why does it give us information? It gives us information because in those metabolic diagrams that I showed you, the arrows went back and forth. And if you think about one pool where 2 arrows are converging on that pool, you're mixing sulfur from 2 other pools that's coming into it. Or if you've got 2 arrows diverging from a pool, then you're unmixing it, or you're essentially distilling the sulfur. And just the predicted value, this here, is based on a single step chemical reaction, okay, and it's very tightly constrained. You don't get significant variations in capital delta 33S. If you have a kinetic reaction, this could be a little bit different, but it also could be very similar, but we're not expecting necessarily large variations, but that's a level that people will and we will investigate. The reason you get the effect is because when you mix pools, the mixtures on the basis of the fraction of 32S that's in A and B weight this way, and the delta variations, even though the units are not the same length for the mixtures, the covariation is linear mixing. So you get a linear relationship, but the reference array is curved. And so we're looking at the deviation for that. And how big is this? If we look at, and these are experiments These are results of experiments from, you mentioned Dave Johnston's work, and then some work that I did. These are experiments for plotting capital delta 33 versus delta 34 of the sulfide, the composition of sulfide normalized to the feed sulfate in a variety of experiments with sulfate reducers. And if you look at this, you notice they're not all at 0, which is, they'd be all between 0 and about 0.02, definitely 0.05 if it were a chemical effect. In fact, they're sitting slightly above here. Okay, these are nowhere near as big as the stratospheric ones, or nowhere near as big as the ones we see in the early Earth environments, but they are measurable and they are reproducible. And this field here is a field that's just, it's a calculated field from a model that takes into account reversibility in the metabolism, and it assigns specific fractionations that largely were accepted for that metabolism, and just shows that the model itself matches the observations when reversibility is included in the model. So this is the evidence for reversibility on a large scale in the metabolism. It doesn't say specifically about where that reversibility is occurring, but you can get to that. And you can actually get to that to some extent. We have a number of steps. So there were a number of steps where there were possible reversibility. We've got 4 isotopes. We could pin down 2 or 3 of those steps independently. With the isotopes, we can't pin them all down. To gain insight into it and to illustrate how one might quantify it, I'm going to go to some work that I did. Actually, I was in France visiting the IPG when I wrote this up. I did this work in Denmark, and we took plugs of sediment, not this sediment, they were more sandy sediment, put them into a plug reactor, flowed sulfate in, a natural population of sulfate reducers, reduced that sulfate and sulfide left the reactor. Okay. We also put an isotopic label on the water, and water and sulfate don't exchange oxygen regularly, so we put an oxygen 17 label on the water, and we monitored the shift in the oxygen isotopic composition of the sulfate that exited the reactor compared to the sulfate that came into the reactor. And this allowed us to look at the fraction of exchange, essentially, if it's assuming it's biological, but that's the only known way to do that, by looking at the oxygen compositions related to the or looking at the oxygen exchanged for the sulfate out of the cell compared to that in the cell. And if we do that, you can calculate out a field for our O17 that we started with. We had a big a 70 per mil label, and for the fraction of sulfate exiting the reactor, you can contour it for fractions of exchange from 0.9 of the 90% of the sulfate coming out of the stuff had been exchanged down to, say, 10% of the sulfate. And these are the data that we had from a variety of different experiments that effectively showed that the fraction of exchange from here coming back this way was about 70% of the sulfate had been through this process. So 70% of the sulfate had been taken up by the cell, come to this point, and then was re-expelled out of the cell, which means it's a pretty inefficient process. And that's one of the questions that came up, is why would organisms be so inefficient? And why would this operate this way? We can also look at the sulfur isotopic data and actually pin down the amount of sulfate that was taken up by the cell and then lost back. So this is just the transport of sulfate into the cell versus what's lost back. And you end up with something on the order of 80 to 96% of the sulfate that's brought in is not actually reduced. Now, if we think about this, this may seem kind of ridiculous and crazy, but if we think about this in terms of this thing, say it was not losing stuff coming back, and say you had a fractionation, an isotope effect of a few per mil from here to here, and then you had large ones that were on these steps down here. If all of it went from here to here, you would transfer a signal from this step back, but then this would go to here and you transfer it to this, but it wouldn't come back to this one. And effectively you wouldn't see any fractionation, yet we see them. So it has to be occurring. So that's sort of the first message, is that the isotopes tell us that it's occurring, and they allow us, they provide a way to quantify that. Now, the situation where we are now is in trying to understand, how do we take a culture experiment and understand what's going on in nature, where you have other things that are forcing the system. And some of the recent studies by Wing and Halevi and others have actually argued that the communities of sulfate reducers will evolve to a lowest sort of energy efficiency state. And so there should be one relationship that they will produce as a function of how they're operating, or one set of rules. And so it shouldn't be as variable. But that also remains to be tested. So that's the argument for reversibility. Now I wanted to go through some of the things that I've been puzzling over and I'm still wrestling with, but they have to do with what happens with these enzymatic transformations. And a lot of it started because I had a phone call from Will Levitt asking to look at some data that he had, and started thinking about what was it that was controlling the fractionation at the enzymatic level, and just wanted to test a hypothesis. But we usually think about this too as a kinetic process, and a lot of times we don't think about them as necessarily being reversible. But they have to be, to some extent, through some of the enzymes at least, to get the signals back. So to go from in that one example from the oxygen, we know that it had to go back through the APS. Well, it didn't have to go through the APS reductase enzyme. I guess it could have short-circuited it, but you'd have to have something that would get past that step. So the question here then is, can you see something in the enzyme itself? And I'm going to go through a little bit about how the enzymes work and what they are. So this is another diagram. A lot of this is done by Oliveira and Piera. They're part of a similar group in Portugal. They have worked with, this is with dissimilatory sulfite reductase. There are other people working on other types of sulfite reductase. Here you have both of the two sides. So you've got a B and a B and an A and an A, and then you've got the two C subunits. And then you've got four centers here, which are the active center. In these cases, it's a cyroheme. It's an iron sulfur electron transfer machinery. Two of them are active and two of them are not for sulfite reduction. And getting into them, there are the channels, and it's not obvious on this diagram, but there are channels that go in to provide access. And this is that diagram I showed you before, with a few more things sort of stuck on here. Here's the whole enzyme. Here's half of it with the ABC. And then here's the channel looking in, sort of blown up with the sulfite. They've got, this is their diagrams, coming into this active center on the cyroheme. And here it is sort of blown up. It's a little bit difficult, but there's an iron in here that the sulfur makes a bond with, and then it docks. And then there's these arginines here and these lysines here. And they're the ones that do a lot of the action. They allow to provide hydrogens and take the oxygens as they're released, and then electrons are supplied from down there. And what typically happens is that on the A and B sites, you typically take the SO3 at docks, it goes to an SO, we'll look at this in a little bit, it goes to an SO2 and then to an SO, and then ultimately it goes to a sulfur, which gets carried off. It can also, these things can also react with the SO3, the sulfite that's there, to make trithionate and thiosulfate. We'll see these are actually going to be important in understanding what the enzyme does. So what Will had done with the group, this is the same group with that had isolated the enzyme and knocked out the C subunit, was they did experiments with the A and B subunits. and they put sulfite in, and then I think they use something like methyl viologen to drive the reaction to activate the enzyme. And what they would see would be sulfite would come in, it would dock into the enzyme, it would go through these steps. It couldn't go out these ways. And then the other sulfite would come in and form trithionate and thiosulfate, and those things would go out into the medium. And they could do a series of experiments and look at how this thing behaved. And the idea was to try to pin down what was the isotope fractionation associated with this enzymatic step. And what they found, and these are the plots, and we'll go through these of their data. The first thing was, and hopefully I've got this yet, it worked, was this is the concentration of the sulfite, that they start with 100% sulfite. Everything's normalized there. They don't have any of this trithionate or thiosulfate. And then this is, well, I guess it's percent versus the fraction of sulfite. So things, the fraction consumed, I don't know why it's not exactly a straight line. This gets consumed, these things build up. And that's basically what happens. This is it's not an obvious ratio. It's a ratio of the thiosulfate to the trithionate on a sulfur basis. So this is not exactly 19% thiosulfate, 80% trithionate. It's something a little bit more weighted towards the thiosulfate than that. So maybe like 25% thiosulfate to trithionate. But it's very constant throughout their reactions. And then this is the isotopic data, and they have the delta 34s and they have the capital delta 33s. And there are a few things here, and that are puzzling and interesting, and sort of raise the questions that we were looking at. This is the sulfite, and it actually changes a little bit with time, going to slightly more negative values. And then the isotopic composition I'll back up here. For thiosulfate, you have an SO3 group. It's like a sulfate, like an SO4, but you replace one of the oxygens with a sulfur. So you've got a reduced sulfur, and then you've got a sulfur that's bound to 3 oxygens in the other sulfur, so it's oxidized. For the trithionate, it is 2 SO3 groups with a sulfur in the middle. And so you've got a reduced sulfur, and you've got 2 oxidized sulfurs. So, and the sulfonate group is the oxidized one, and the reduced ones are the sulfane group, essentially. I will refer to it that way. It's a little more complex than that. They were able to analyze the oxidized and the reduced, so this is a combined oxidized from trithionate plus thiosulfate and reduced from trithionate plus thiosulfate. But the difference between the two was a relatively constant, about 16 per mil. Okay, and it was smaller than what would have been predicted on the basis of partition coefficients between the products and reactants. And that's one of the things their finding found, was that this was smaller, and people have run with that. The question then is, can it be larger than that? The other thing that they found, and this is moving down to this capital delta 33, is that there were differences in the capital delta 33 between the reduced group and the oxidized group, and then this is basically where the sulfite would have started. It evolved up this way with the experiment, and this difference also needs to be explained. And their argument was, well, since it's non-zero, it can't be equilibrium fractionation. Maybe it's a kinetic isotope effect. And the question that I wanted to ask was, well, could this be reversibility? And can we test that? And we can go through that by looking a little bit at how this enzyme works. So here's this cyroheme here. You've got a whole bunch of carboxylic groups, and this is the sulfite that's bound here. The iron is here. And basically the steps that it goes through is you've got these arginines, and they will connect up with the oxygens, and they'll basically pluck them off, going from an SO3 that's docked to the Fe, and then going to an SO2 that's docked to the Fe. These are the lysines here working on this one, and then going to an SO, and then ultimately to an S2. And then this one might react with another sulfite in their experiment to make a thiosulfate precursor. It's attached to an Fe here. This one might react ultimately to make this trithionate. And if it's a reversible system, then we're not necessarily dealing strictly with a kinetic reaction pathway where you're going over a potential barrier, because you've got forward and backward reaction going on, and you can look at the partition functions for these various states in between, or these steps in between. And so what we decided to do, we can do this, is to go through, build a model, try to jump in to try to see if we can figure out what the partition functions could be, and then to see if those partition functions with the mixing model can explain some of the observations. So this is, in order to do that, I only have so much computing power, so I have to truncate things. I think we're using like 500 gigabytes of RAM and 4 terabytes of swapping space to do this. But we basically have to truncate it. And then you have the sulfite here. This is from the structure that they provide. And then you truncate it, and you have to set up and try to understand what the charges should be, what the spin state should be, and then go through that calculation and determine what is the partition function for this species. And the way that we've done this is we've locked everything in place to basically set up a force field, and we've allowed the iron to move, the sulfur, and the two oxygen, or the nearest oxygens to move, to get an idea of sort of the first order partitioning that would exist. And then using those partition coefficients, we can then go through and look at the look at whether this will work. And so this is we're using a density functional theory of B3LYP, and this is the basis set. I guess I should have put that up there. It's a truncated structure. We've got this longer range order. We froze atoms in the far field basically to make this electronic field in which these other things will move. And then we used a multi-level, this is technique to assign multiplicity and to set up the calculation. We can check our result when you go through, you optimize things, you can check it against the structure that exists. And so these are the two structures overlaying the original structure and the calculated structure. And you see here the oxygens moved a little bit by about 0.3 angstroms, the sulfur moved by about 0.06, and the iron down here, which I guess it's that one, moved by about 0.06 angstroms. So it's very consistent with the structure that existed. This thing did not sort of helicopter around and end up in a very different environment. And I think I'm still finding people to talk to make sure that I'm doing this correctly. But the results, basically, you can calculate out the partition functions for the different isotopes for each of these different species, and you can then use them to determine the partitioning of sulfur between, say, this species where you've have a docked SO3 on the enzyme, and this species here with this S here, okay? And this is what we've done here, and put it through a model where we've assumed that these are reversible, so that we're dealing with those things. And the model that we would calculate would give us a field of compositions that would look something like this, okay? This is the field of fractionations between what they measured between this and this species here. And so I would argue, and this shows, also it shows the difference in this capital delta 33, so, and for 34. So my argument is that at least from what we see, we are fully consistent with the enzyme steps being reversible if the conditions are allowed to allow it to go in reverse. Now there will be a push as you're feeding the enzymes with electrons to make them go in one direction or another. And so these effects need not be something that's kinetic. It could just be something that is fully reversible. Now, everything is not completely solved, and we're still trying to work this out. This, if you remember on that other diagram, we had and this now I've normalized to the starting sulfite. You find the starting sulfite was similar to this reduced sulfur and different from this from the sulfonate group. And if you combine these two, which is the net stuff that's produced, you get something here. And there's a difference here from there to there that needs to be understood. Now, we do see that this difference, if you put it into Rayleigh equation, would explain this behavior going up this way. The behavior in that model could explain the behavior of this thing going down this way, but this difference still needs to be understood. And part of this could potentially be related to the speciation of these of the sulfite in the free cytoplasm and the speciation as those sulfite or bisulfite goes into the channel. Because if you have it in a free fluid, the speciation can be shifted somewhat, or you could change the pH somewhat. And I want to go through that next, and I think we've got time to do that. So the next two examples are two sort of steps. The first two were the issue of reversibility, and then what potentially happens on these enzymes. And just to be clear, I'm not trying to rule out the possibility of there being kinetic isotope effects in the enzymatic transformations. There clearly are for unidirectional processes. But I am trying to say that if there is reversibility, which I think there likely is, or we have to find another way then to explain these capital deltas, the reversibility that we can predict based upon a model that takes into account reversibility and exchange between the different parts of the enzyme, or the different steps of the reduction in the enzyme, can account for a lot of the observations. In addition to that, we also have this issue of protonation. So you can add hydrogen. You don't really add it unless you're under really acidic conditions to these guys. But bisulfite and H2S, or sulfide, you can add it to. And I'm going to just show you quickly. For sulfide, it's not a huge effect, but it illustrates some things nicely. This is sulfate, low pH. This is sulfide from bisulfide to H2S. And then this is where you go bisulfite to sulfite, both in that critical pH range. The key things with this is that the pH range of the acid dissociation or the protonation, the constant, changes as a function of temperature. Okay, this is for sulfide, and this is for sulfite, and these things you can calculate. And each of those species, say for the HS, it has the sulfur is bound to a single H, and it essentially dissipates its energy that way. For H2S, it's bound to two H's, and it dissipates its energy that way. This affects the partition functions. And the same thing with HSO3 versus SO3 2-. And you can take that and you can put that into this, and you'd say, okay, H2S versus HS- as a function of temperature, you'd expect it to be, looks like it's about, say 1, 2, 3 per mil fractionation at 0. These are calculations that Daniel Eldridge did for his PhD, and then at higher temperatures it's somewhat smaller. But the neutral pH, or the pH of water, as you change the pH for this, you change the speciation dramatically between these things. And so at pH 7, the composition of the sulfide, the net composition, would be something like this, and then it would go down like this. And this would be relative, this would say be total sulfide, and you could compare that to some other species. This is straight equilibrium calculated. Yeah. This is straight equilibrium. But you can see how you could change the behavior by a per mil or two. These would be the fractionations that you would see. Now it gets a little bit more interesting when you deal with bisulfite, the SO3, and you protonate it, because you could have the sulfur here bound to the 3 oxygens, and you can put the proton on one of the oxygens here, or you can put the proton on the sulfur here. If you put the proton on the sulfur here, it adds a bond here, and the sulfur actually ends up being more strongly bound. So it has a dramatic effect on the partition coefficient of this versus sulfite without the protonation. This one also is different than the sulfite without the proton, because the proton weakens this bond and then makes this the partitioning slightly different there. Now, how much hydrogen goes here versus how much hydrogen goes here is something that actually has an effect on the fractionation that's observed for the sulfite. And that partition, most of it, about 75% at 25 degrees is here, 25% is here, but this difference is about 20 per mil. And if you look at this as a function of let's see, this is actually this thing also can vary as a function of ionic strength. These are some calculations that Daniel had done. You can see that the speciation of these different, the HSO3 here, where it's hydrogen on the sulfur, gets higher at low ionic strength and lower at higher ionic strength, and vice versa here. So this thing varies, and with a 20 per mil difference between this species and this species, the net effect of this on the total dissolved S4 is significant relative to the other species. How he did the calculation, just quickly, these are calculations that were done in water clusters. In this case, everything moved, and it's an averaging, it's essentially an averaging technique to deal with the interactions between the water molecules and these things that appears to be much better for constraining the isotopic, or the things that control the isotopic fractionations. So he did the calculations. He also afterwards looked at the speciation with NMR techniques. This is after he left Maryland working at Carnegie, and he was doing this in part to explain weird behavior that was seen in experiments. So this is a study from the 1970s, and I don't know, that's Truva Erickson or how to pronounce that, but something like that. This is the fractionation factor. This would be about 10 per mil. This is 0 per mil. This is temperatures in Kelvin. And these were some experiments that they had done, and they found basically that there was no dependence on temperature between, I think it's total sulfite aqueous and SO2 gas. And so they saw this weird temperature dependence. And if he takes into account the temperature dependence of this isomerization, this is what he gets calculated by looking at the calculate partition function for this HSO3 and this OHSO2 here gets this, and essentially it goes right through the data. So I think this is actually, it's an important consideration that has to be taken into account, and he has been raising the point that changes in the speciation, either as a function of temperature or potentially as a function of environment, could actually account for some of the variations. And I guess I must have saved the slightly wrong version, because I did a version on the train coming up. The shift between the sulfite that's out in the media and assuming that the sulfite that's in the channel gets buffered by the reaction of about 0.03 per mil is consistent, or is almost fully accounted for by the shifts in this speciation. If, for instance, the pH was different inside the enzyme as opposed to outside, or if there was something controlling the protonation inside versus outside. So the question then is, why is it useful, and why do we want to do this? And the reason is because if we understand how the mechanics of these different organisms operate, we can say things that can be used to interpret and understand whether changes in these, say, the difference between sulfate and sulfide in the environment reflect something to do with the organism. And if they reflect something to do with the organism, then maybe we can get back and say more about the environment. So sort of to end, I think the considerations we want to do, we've got a long way to go. Understand the metabolic structure and the pathways play a critical role in how the fractionations are produced. The mass transfer and reversibility is also critical in terms of the magnitude and leaving signatures behind. I'd say in addition to kinetics, we've got partitioning and reversibility and speciation that also can play a role. And I think we have evidence for all of these. I don't think we've got the metabolism sorted out, but those are the types of things that I would look for. So, thank you.
Okay, thank you. Thank you, James. Any question? Lisa, I was sure you would ask.
Well, I might ask a stupid question.
Go ahead.
Okay, I don't know anything about this topic. I'm just thinking, so a lot of the things that you were showing, like the fractionations with the speciation and the pH and the temperature, these were uncatalyzed reactions.
So the all of the lines on the plots were calculated lines, and the inputs to calculate those were done through ab initio calculations of partition functions, which are essentially thermodynamic quantities for isotopes.
Mm-hmm.
And so they assume that equilibrium applies, or they assume that there's energy minimization for them to apply.
Mm-hmm.
So there's no catalysis in this.
Yeah.
Catalysis is a whole nother level.
Yeah, because that's I guess that was what I was trying to understand. I'm more looking at it from the perspective of minds, because usually how I try to understand how organisms or enzymes are doing things in the environment is, you need to know you need to put in all your uncatalyzed information there and all the information that you know on all of these other fractionations, and then you're looking at what's left and if there's any differences caused by the biology of an organism, or pushing the organism into one state or another, or adding an inhibitor, if there's something else in the environment that's changing this. So I don't know this enzyme, so I just wanted to clarify that I guess we're not at the stage yet of really knowing. What did you want to say?
So I think I can answer some things.
Mm-hmm.
I think we're at the stage where we can say an awful lot.
Mm-hmm.
And I think just to show you quickly, if we take a step like, I turn this on. Take a step like we'll say like this one here. Mm-hmm. Here you've got a site that's interacting with this oxygen, a site that's interacting with this oxygen here. And these things, and you've got something here off of this amino acid chain that's interacting with this oxygen here. Those things are changing the electronic environment around those two oxygens. You've also got electrons coming up this way through this cyroheme. Mm-hmm. And effectively that's the catalysis. And that is occurring, mm-hmm. and it's inescapable. And that drives the change from here to here, okay? Mm-hmm. Now, all that I'm doing is I'm saying, okay, say you're going from here to here, but you're also coming back. Mm-hmm. Is it the step that goes from here to here that matters? Or if you're going back and forth, then your rates, you're no longer talking about a forward rate versus a no rate going backwards. You're talking about something that involves this state versus this state. Mm-hmm. And so the question that I asked was, could we explain the observations with the isotopes that we see by considering these states separately, and therefore that these things were reversible. So there would be catalysis, but it would not be the only thing that was going on. It may not even be the first order control on the isotopic effects that we're seeing in the experiments. And we can use the experiments to place a constraint on how much went backwards. And we require about 60% to made it back. That means that you've got a lot more. You've got 100 forward, 60 back.
Yeah, because in your bioreactor experiments, you're feeding in and you're looking out, and then you're...
Yeah, that's a different experiment. Yes.
That's like a community in there, or
That's
is that...
Oh, that was a quite crazy wild community, yes.
Okay, yeah.
Yeah.
And so that... Okay, I can understand you're trying to understand this, you know, how these shifts are occurring in communities in the real world. And... so I guess, do we know all the enzymes? Are we pretty sure we know all the...
I think for this organism, I think my understanding is that they're pretty well constrained. For these pathways. I mean, they have been sequenced. They also have isolated the enzymes. They've crystallized them. They know that they're different classes and different groups of organisms. So I think a lot of the biology has been worked out.
Okay.
Hi, James. So, do you have any idea why you have this increase in the fractionation between sulfate and sulfite over the time? Now that you have all this data, do you have any idea where the difference can come?
So the difference that you're talking about is sort of the thing on that this slide. There are two hypotheses. When we originally did this, we thought that there was a change in ecology structure that was happening somewhere here. So it was actually not necessarily a change at the organism level. And we thought we could explain it that way. More recently, Will Levitt and Dave Johnston, who did the other work, did a series of experiments where they saw a dependence of the isotope effect on the rate of sulfate reduction, which had to do with how much electron donors were available, so organic matter for the sulfate reducers, or the quality to live. And in fact, with the models I said showed there were supposed to be consistent relationships between 34 and 33 because of these evolution type experiments. They also came to a similar conclusion. And the idea there is that there was some environmental change that had to do with not with the population structure or the structure of the environment, which is what we had proposed, but actually had to do with the organisms themselves and how they were responding to the environment. And so they would say that sometime about 300 million years ago, there was something that changed with the oceans that allowed for these fractionations to be expressed. And then sometime, this could be an environmental control, but another type of environmental control, but we're getting into little data back here. But this thing they think is related to the organic matter that is available for the organisms. But it's not known.
Because these are looking, I mean, are these from like these cores that you're taking out of sediments? And
this is a huge data compilation that people had made. And so the first thing, this is sulfate. This is sulfate shifted by 60 per mil, and you see it matches about the bottom of this line here. And in the 1990s, it was thought, okay, this thing just matched, and this was always constant. And then we went in and we did an average through this and said, okay, we get a difference. Is this real? And then we went through and we did a model that took into account the 33 sulfur that was independent of the assumptions of the averaging, and basically got a line that fit right through here. So we said, okay, in fact, we've got two things now that seem to point to the same thing. So we said, okay, we think it is real. Then we tried to explain it. But
for instance, if you took a core, a sediment,
it would have a huge variability.
And so it was 0 to 800 years. And if you were to slice these up, you would be able to look at the community that was still alive?
These are these are rocks.
Oh, these are rocks.
These are rocks.
They're not
And-
like mud.
And
I mean,
if you look here, this is no.
just asking
This
these
is
questions.
a punctuated record, and probably this line
Okay.
here is one formation,
okay.
or would be one core, and then you gap, and then here's another core, and you see at any one time you get this whole range. Why the average should come through and give some consistent story is
Okay, so how
is
do you make the
mind-boggling.
link to the community structure of the organisms? I guess that's
So we made the link by looking at the sulfur 33 and looking at how different types of metabolisms affected it, and then we looked at the role of reoxidation versus sulfate reduction, and so we would have had a break in terms of things being more oxidized from here to here and less oxidized from here to here. Okay. But that was the way that the global oceanic model responded
okay.
when inverted, and the Harvard group and Baz Wing and Itai Halevi looked at the response of the organisms and said, okay, the organisms in principle also could explain this.
So yeah, going at the extreme scale, at the level of the cell, do you have any idea where the reaction occurs? I mean, in the cytoplasm of the cell, or do you,
So-
and yeah, another question or comment. When people are doing cell experiments, they work on the bulk of the cell, or they try to separate the cells compartments.
So these are prokaryotes, and so they don't have a nucleus. The ones that do the sulfate
Okay.
reduction. Most of the chemistry occurs in the cytoplasm, but there are those. The way that I envision and the way I understand it is there are these enzymes that are floating around, and many of them. The sulfite reductase is relatively large, but there still would be many copies
Okay.
of it. And then the chemistry is sort of an ensemble of it moving through, and some of it happens in the cell, some happens inside an enzyme, some happens on the cell wall.
It's tricky.
It's not a single reaction that's going on.
Okay. Okay.
Um,
Do you know if some people have tried to play with mitochondria, because mitochondria is full of sulfur, and there is a lot of reaction, very funny reaction into mitochondria that can explain some of the observations.
I'm sure there are people who have. I haven't,
Okay.
and I don't know, but I'm now curious to look and learn more.
James, there is one thing that I would like to understand. So you, in your original papers, where you run microbial sulfate reducers, you develop the concept that everything can be explained by reversibility, which allow you to make actually simple calculation and mass balance and to discuss the different models that were published before. And when you start questioning reversibility, as you've done it in your presentation, of course, we all think in terms of variation in temperature to address if it's reversible or not. And then you move in and say, well, it's hard to test the dependence upon temperature with reversibility because we may change the speciation by a lot, correct?
You may yes, you may change
Yeah.
in a model, in testing reversibility, we need to know what the isotope effect is going from here to this pathway or from here to this pathway. And if those change, then it gets more complicated.
And therefore, when if you move back to your original experiments, the variation in delta 34 and capital 33, was that related to a change in the various pathway, or was it simply a change in the speciation?
So I think if I divide things up into sort of the first order effect, the thing that controls everything, then the second order effect and things like that. In those initial views were pretty coarse, but I think they still hold at their level, because they're dealing sort of with an ensemble effect, counting all of these species going to all of these species, and all of these going through this to all of these, and so I think within the structure that exists, they still work. But certainly, if you go and you try to take them apart in more detail and say, okay, can we use the information that we have to track what happened here and here and here and here and here? There's no way that we can do that, because we all of a sudden have too many knobs to turn and only so many, or there are too many variables and not enough constraints. And the constraints we have are the 3 sulfur measurements, and if you add oxygen, a 4th. And then if you had concentration, you could have a 5th or something else. But you've got many more than that here.
So how far are you from taking the curve of Cornfield and others and tell if it's a change in the type of the microbes, or versus change in environmental conditions that will drive different
I think
pH or things like that?
I think the argument about the change in the behavior of the microbes was plausible. It was really nicely done. So I think it's a valid one. The change in the ecology or community, also maybe, I think that also still could hold. It would require a different set of assumptions. I think it's going to take a while to know which is the right answer. I don't think at this point we can rule them out.
Yes. In your ab initio calculation,
Yes.
I don't well understand the shape of the.
Is it the? Oh, the shape of the field?
Yeah, the shape of the field.
Okay.
Why there is that point at -15, for instance?
So these, well, let me just go through. These are the experiments that they did at 2 temperatures. So this is 30 degrees, this is 25 degrees, and this is the fractionation between the sulfite and the reduced group. This thing here and this thing here is the calculation that you get when you put a fractionation from here to here, from here to here, from here to here, and from there to there. And then you calculate it through this reaction network. The way to look at it is this upper curve, the thing that's here, is if everything is fully reversible and you're just varying the amount coming in and out here. And then these steps coming back basically relate to the reversibility on each of these steps here. And what happens is if you vary things consistently, you basically end up filling in this field because the field retracts. So if this was to here, it would be truncated here and it would come around.
Yeah, that's the truncated, the truncation.
Yeah. But it's the
what? Why the reverse
shape
is truncated?
Why does it go like this? So when you have mixing, the mixing puts curves like this. Okay. But in this case, the species that we measure are actually on those curves. So we're sort of moving things around, and it basically does a mirror image of the curves. And so then you get curves that go like this. And these are basically related to mixing relationships along each of these steps. So as this thing goes from 0 to 100 backwards, it may go like that and so forth. Okay. So this is the compositional field that is allowed by this structure assigning the fractionations that would be controlled by these intermediate states. Okay. That accepts that they're calculated correctly. I don't think they're that far off, because as you move further and further away, the effect on isotopic partitioning diminishes
So
only little.
if I understand well, the position of the truncation points is a function also of the initial composition.
This field here
Yeah,
is
moving.
anchored to an initial composition here with this coming in. If
So-
you move the initial composition, this
Yeah,
field
you
would
move.
move.
You move the field.
You could translate the
okay.
field.
Another question? Please.
Clarify something. Your work was done mainly on soil, organisms in soil through cores, and on rocks, cores and rocks too.
So the organisms that we studied, we get from the various organism libraries. Okay, and those organisms have been cultured. Some are ocean sediment. Some are from ditches behind the MPI and Bremen, stuff like that. The analyses involved rocks. A lot of the things here were just calculations that were based upon ab initio methods.